5.
Question 5. The Galapagos are a group of islands 600 to 800 miles off the coast of South America. Less than 3 million years ago, some finches of a single ancestral species reached these islands from South America and bred successfully. The number of islands has increased over time, further isolating groups of finches. Use this information to name the type of isolation barrier involved in the speciation of these finches and explain its role. Okay, number of islands has increased over time, so what we have is a geographical isolation barrier, which has the same reason as every other barrier. Okay, what we have here is it prevents gene flow. Okay, so it stops interbreeding between the populations. That is what is essential. Okay, you then got a table of information. The estimated number of islands and finch species in the Galapagos over the last 2.8 million years. So this is obviously present day, and this is working back the way. And so our number of islands in the group has increased as we've gone, although it stayed the same for the last 500,000 years. The number of finch species has increased throughout. Okay. Calculate the average increase in the number of finch species per million years over this entire period. Right, so I'm looking for species per million years. Our number of species is 14 in 2.8 million years. So 14 divided by 2.8 gives me 5. That's the answer. Okay. Suggest what could have caused the change in the number of finch species over the last 500,000 years. Okay, so what we know is that the number of islands has not increased, but the number of finches has. So, sorry, finch species, not necessarily the number of finches. So if it's not the number of islands, it's no longer geographical. So I could have behavioural isolation, or I could have had an ecological isolation. That'll give you the mark. Or you could have been nice and posh and gone for sympatric speciation, because that's really what those are leading to. Okay, a molecular clock was used to estimate the dates of divergence of each species. Give one example of the type of data required to generate a molecular clock. Okay, so a molecular clock is you looking at the changes within a sequence of a particular gene. So I think the obvious one to go for is that I need sequence data for the genes. What I could then absolutely need to know is that if I'm going to have a clock, I need a rate. So I need a rate of mutation to allow me to figure out how far that sequence has diverged, given how often you get a change in the sequence. And that's the question.